Dear viewers, greetings. In this video, we are going to discuss about the ion exchange chromatography. The contents of this video includes introduction about ion exchange chromatography, principle of ion exchange chromatography, types of ion exchange chromatography, types of ion exchange resins, instrumentation of ion exchange chromatography, steps in ion exchange chromatography, factors affecting ion exchange chromatography, applications of ion exchange chromatography, advantages of ion exchange chromatography, and finally, limitations of ion exchange chromatography. Ion exchange chromatography. Ion exchange chromatography is a type of chromatographic technique used for the separation of charged ions. Ion exchange chromatography media contains negatively or positively charged functional groups which are covalently bound to a solid support, yielding either a cation or anion exchanger respectively. Any charged substances like amino acids, proteins, and nucleic acids are purified by ion exchange chromatography. In 1975, Hamish Small, Timothy S. Stevens, and William C. Bowman introduced ion exchange chromatography in modern form. Principle of ion exchange chromatography. Ion exchange chromatography separates the molecules based on the charge on each molecule. Based on ionic interactions, ion exchange chromatography keeps allyl molecules on the column. Positively and negatively charged ions make up the ion exchange chromatography matrix or stationary phase matrix. On the stationary phase matrix, molecules engage in electrostatic force of attraction with opposite charges. Immobile matrices with charged ionizable function group are present in the stationary phase. Alkalis, acids, and buffers are used as suitable solvents or mobile phase. Organic solvents are less useful, so they are not used anymore. These ionic functional groups interact with analyte ions of opposite charge, and the ionizable molecules are retained or eluted based on their charge. Types of ion exchange chromatography. There are two types of ion exchange chromatography. They are anion exchange chromatography and cation exchange chromatography. The first type is anion exchange chromatography. Anion exchange chromatography is the separation technique for negatively charged molecules by their interaction with the positively charged stationary phase in the form of ion exchange resins. It is also called as acidic ion exchange. The second type is cation exchange chromatography. Cation exchange chromatography is the separation technique for positively charged molecules by their interaction with negatively charged stationary phase in the form of ion exchange resins. It is also called as basic ion exchange. Types of ion exchange resins. Two main groups of materials are used to prepare ion exchange resins. They are polystyrene and cellulose. Resins made from these materials differ in their flow properties, ion accessibility and chemicals, and mechanical stability. Polystyrene resins are proposed by the polymerization reaction of styrene and divinyl benzene. A high concentration of divinyl benzene produces high cross linkages. Polystyrene resins are very useful for separating small molecular weight compounds. Cellulose resins have much greater permeability to macromolecular polyelectrolytes and possess a much lower charge density as compared to the polystyrene exchangers. It includes carboxymethyl cellulose acts as cationic exchanger and DEAE cellulose acts as an anionic exchanger. Instrumentation of ion exchange chromatography. The important parts of the ion exchange chromatography are pump, injector, column, suppressor, detector, and data processor. The first part of the ion exchange chromatography is pump. The pump is considered to be one of the most important component in the system. The pump provides a continuous constant flow of the eluent through the injector, column, and detector. The second part of the ion exchange chromatography is injector. Sample introduction can be accomplished in various ways. The simplest method is to use an injection valve. Liquid samples may be injected directly, and solid samples need only to be dissolved in an appropriate solvent. Injector should provide the possibility of injecting the liquid sample within the range of 0.1 to 100 ml of volume with high reproducibility and high temperature and high pressure up to 4000 psi. The third part of the ion exchange chromatography is column. The smooth flow of the ion exchange chromatography is based on the column. The length of the column should be around 3 cm to 50 cm depending upon its purpose, and the diameter should be between 2 mm to 5 cm. The column material might vary from glass, stainless steel, inert plastic, or titanium. The fourth part of ion exchange chromatography is suppressor. The suppressor reduces the background conductivity of the chemicals used to elute samples from the ion exchange column, which improves the conductivity measurement of the ions being tested. Suppressors are membrane-based devices which are designed to convert the ionic elute to water as a mean of enhancing the sensitivity. The fifth part of the ion exchange chromatography is detector. The most commonly used detector in ion exchange chromatography is the electrical conductivity detector. The sixth part of the ion exchange chromatography is data processor. In routine analysis, where no automation is needed, a pre-programmed computing integrator may be sufficient. For higher control levels, a more intelligent device is necessary. They are data station or mini computer. Steps in ion exchange chromatography. Ion exchange chromatography separations are carried out mainly in columns which are packed with an ion exchanger. These ion exchangers are commercially available and they are made up of styrene and divinyl benzene. Example, DAE cellulose is an anionic exchanger and carboxymethyl cellulose is a cationic exchanger. The choice of the exchanger depends upon the charge of the particles to be separated. To separate anions, anionic exchanger is used, and to separate cations, cationic exchanger is used. First, the column is filled with ion exchanger. Then the sample is applied, followed by the buffer. The tris buffer, pyridine buffer, acetate buffer, citrate buffer, and phosphate buffers are widely used. The particles which have affinity for ion exchanges will come down the column along with the buffers. In next step, corresponding buffers separates the tightly bound particles, and finally these particles are analyzed spectroscopically. Factors affecting ion exchange chromatography. Usually, four factors affects the ion exchange chromatography. They are size of the ion, valence of ion, concentration, and finally, nature and properties of ion exchange resins. The first factor is size of the ion. The exchange of ion increases with a decrease in the size of the ion for similarly charged ions. The second factor is valence of ions. The exchange of ions increases with the increase in valence electrons. Third one is concentration. The more the concentration, the more is the exchange of ions. And finally, nature and properties of ion exchange resins. cross-linking and swelling are two important factors that affect the ion exchange chromatography. When there is more cross-linking, then it swells less. So, it has less pore size, making it difficult to exchange ions of larger size molecules. Applications of ion exchange chromatography. Ion exchange chromatography is used for the separation of amino acids and proteins. Ion exchange chromatography is used in softening of water. The compounds of pharmaceutical products can be separated by ion exchange chromatography. Ion exchange chromatography is used for the separation of similar ions. Ion exchange chromatography is involved in complete demineralization of water or removal of cations and anions. Ion exchange chromatography is used in the discovery of trans plutonium elements in the actinide series. And finally, ion exchange chromatography is used in the separation of borate complexes of sugars. Advantages of ion exchange chromatography. Ion exchange chromatography is one of the most efficient methods for the separation of charged particles. Ion exchange chromatography can be used for almost any kind of charged molecules, including large proteins, small nucleotides, and amino acids. Ion exchange chromatography is used for both analytical and preparative purpose in the laboratory, the analytical uses being the more common. Inorganic ions also can be separated by ion exchange chromatography. Finally, limitations of ion exchange chromatography. There are two limitations. The first limitation is only charged molecules can be separated, and the second limitation is buffer requirement. Dear viewers, that's all about the ion exchange chromatography. Thank you for your support. Thank you.